It is. What's poppin'? We caught a vibe over here. One of us is a really cool plant and the other one is a really cool mushroom. A little funky, but quite frankly, we are built the same, at least at a cellular level. All the eukaryotes, we be racking the same core cell structures because of some sort of ancient group chat that we never left. That ain't no coincidence. There are some ancestral receipts. Here's the tea. We're going to be spilling everything that we've been talking about in section 7.7, which is all about common ancestry. Dude, we're going to talk about our glow up from our single-celled great ancestor. Let's slay, but before we get started, make sure to note the disclaimer right here.
I want you to take a look outside your window. If it's the summertime, you might be seeing some various different birds or different trees, and you might be shocked to know that they're related. Yes, I said that, they're related. See, they're all eukaryotes, and what eukaryotes share is the same basic cell structure and function. And we all believe, in terms of biology, that while we are very different, fundamentally we have the same common ancestor, at least according to AP biology. And this all stems from the endosymbiotic theory. The endosymbiotic theory, while not covered in this unit, will help you understand when we talk about the different structural and functional evidence as it relates to eukaryotes. Here's the thing. The endosymbiotic theory is a interesting theory, as it states that once upon a time, a long time ago, there was a world with no oxygen. And all the organisms that lived on this planet were able to sustain itself without oxygen. But then, out of nowhere, there was oxygen. And prokaryotes, which were once seen as the primary source of a cell, or seen as the primary cell per se, were found to be engulfed by another cell, which we now deem eukaryotes. See, what happened was we believe that mitochondria were able to sustain the oxygen that was in the environment. And the mitochondria itself has its own DNA, fun fact. And another fun fact, we attain our DNA from our mother. Did you know that? Well, now you do. But for now, you need to understand, in the endosymbiotic theory, the mitochondria was engulfed by a cell, which ended up becoming what we now know as eukaryotes today. And mitochondria is a interesting organelle, as it creates its own ATP, or energy, and is able to sustain itself in its location within the cell. So this is a reason why we've developed the endosymbiotic theory. Before we focus in on further learning objectives, I just wanted to point this out. Now, for the AP biology exam or any college biology exam, you might not see the endosymbiotic theory per se being asked as a question, but it helps create a picture of how important eukaryotic organelles are in terms of how they've evolved over a period of time. Let's talk about the structural and functional evidence as it relates to this idea of common ancestry. If we take a look at structural evidence such as membrane-bound organelles, linear chromosomes, or cytoskeleton, these three structural features support this idea that we came from a common ancestor. If you take a look at organelles within a cell, such as mitochondria, ER, nucleus, cell membrane, and Golgi, eukaryotic cells have these membrane-bound organelles, and they have very specific functions across multiple species as long as they're eukaryotes. And this allows us to realize that because they have similar functions across multiple species, say yourself and a monkey, that leads us to believe that there could be some structural evidence relating to common ancestry. Let's talk about linear chromosomes. This is a picture of eukaryotic DNA, and this is a picture of bacterial DNA. Bacterial DNA is circular. However, when we talk about eukaryotes, we notice that the DNA double helix, that if we unwind and we take a look at the chromosomes themselves, they have linear chromosomes. And this is very dissimilar to bacterial DNA, which is circular, which leads us to believe that this is also structural evidence. Let's also talk about the cytoskeleton. The cytoskeleton wraps around the cell and actually holds the cell's shape, allows for movement, and allows for cell division. That cytoskeleton is composed of what's called microfilaments and microtubules. These two structures are the smallest to the largest. So you usually have microfilaments, intermediate filaments, and microtubules, but microfilaments and microtubules really make up the cytoskeleton. And if we take a look at a lot of the cytoskeleton in multiple different types of cells, you could see similarities in the structural shape. Now let's take a look at functional evidence, or the way things move or react. The process of DNA replication is the idea of taking DNA and replicating itself. Also, DNA, when it is replicating, has to repair. And what we do notice for eukaryotes is they use very similar enzymes, such as DNA polymerase, to really copy the DNA, or they have repair-related enzymes to fix the DNA as well. When we talk about gene expression, such as the process of transcription and translation, which leads to the central dogma, which tells us that DNA becomes RNA, which eventually turns to protein, and who we really are, our bodies have all different types of genes, and whether or not they're turned on or off is the same across multiple different species. The process of DNA polymerase acting to help assist, and then splicing and ribosomes are also additional evidence to show that what we have in ourselves is very similar to other species, which could lead to this idea that it could be evidence for common ancestry. We talked about this earlier, but we also talked about the endosymbiotic theory, which is this idea that very evolved structures such as the mitochondria or the chloroplasts have its own DNA and have its own functionality, which gives this idea that they also produce their own energy outside of the cell, but they are within the cell. So that also gives us functional evidence of evolution in of itself. When we talk about membrane-bound organelles, we often talk about eukaryotic cells, as previously indicated. And when we're talking about these eukaryotic cells, we're talking about this idea that the key organelles that are membrane-bound play an intricate function into the development of the cell. So, for example, we know that nucleuses, which is the center here of the cell, basically stores and protects DNA. Organelles like the mitochondria are ones that produce energy. And we talked about the endoplasmic reticulum and the Golgi apparatus. And basically, these are involved in protein and lipid packaging and transport. And these are interesting because even though each cell is unique, they carry the same patterns throughout the cell, regardless if the cell is in a human or in a different animal like a bug. Because they're all eukaryotic cells, intricately, they're very similar in fashion. We do know that prokaryotic cells lack these internal structures. That means or leads us to believe that they're less evolved and that eukaryotes suggest that they have some sort of origin that relates to those prokaryotic cells, which is how we came to that theory of the endosymbiotic theory. Let's talk about linear chromosomes. Now, remember, in the nucleus, we have our chromosomes, and in eukaryotic DNA, these chromosomes formulate such a shape that's linear. They're not circular in shape like bacteria, and they're stored in the nucleus, and they have these ends that are protected called telomeres. Once again, the structure and the functionality of these linear chromosomes are similar across various eukaryotes, which indicates that there's a common ancestor essentially. Let's talk about introns. Now, if you've covered this in an earlier unit, this might be a review, but basically there's a process before translation, and it's a process in which we have genes that have what we call exons and introns. So EXON are exons, and then we have introns. And what happens with the introns is they're actually spliced out or they're removed through RNA splicing. So the process you see here is RNA splicing. And essentially what's happening is when we're going from DNA to RNA, there's a process before RNA goes to protein, which is called RNA splicing. And this is key for a lot of eukaryotic RNA processing. It's a very complex process in which essentially what happens is you have what are called introns and exons. And they often go, I'll call these EI exons or introns. And then what happens is the introns are then removed, and then the exons come together essentially. And so what ends up happening is that is what's left over. So that process itself of basically having introns being removed is a unique process of eukaryotes. Prokaryotic genes rarely have those introns. Once again, basically further explaining how prokaryotes are less evolved or less developed than eukaryotic genes, and essentially giving proof for an evolutionary origin. Let's try to fix or understand the following question. If you need a moment, take a moment to pause, and then we'll try to see if we got the correct answer. Okay, so it says, what statement best supports the endosymbiotic theory regarding the mitochondria and chloroplasts? So regarding mitochondria and chloroplasts. And it says, A, they are found in all types of cells. They have single membranes. They contain their own DNA and are double membranes. They evolved independently in different organisms. So the best answer choice here is C. Let's talk about why the other responses are not favorable. So they're found in all types of cells. Look at the word all. All is an extreme statement. And for most AP biology questions, you want to stay away from these extreme answers. Remember, the endosymbiotic theory says the eukaryotic cell has evolved from at some point from the prokaryotic cell that was engulfed and eventually engulfed a mitochondria-like structure that may have been manipulated. So they're not found, essentially mitochondria and chloroplasts, in prokaryotes. So this would not be a good statement. They have single membranes. Remember, mitochondria and chloroplasts are actually double membraned. And if you covered it in an earlier unit related to cells, you'd see that. But you just need to know they are membrane bound and they actually have multiple membranes. And then C, they contain its own DNA and are double membrane. Yes, they're double membrane and they contain their own DNA. Let's take a look at D. They evolve independently in different organisms. Now, we do know that mitochondria and chloroplasts have evolved, but even though they evolved, they're not evolving independently from different organisms. They're sort of dependent on their environment in which they live in, right? And that's how mutations come about. So that's not the best answer. So we don't really like it for this type of question. I hope this video helps you understand section 7.7. I hope that you take the time to hit that like button, hit that subscribe button. Make sure to check out our AP Biology playlist for this year, so that way you can evolve into understanding better material for the AP Biology exam or college biology exam. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.